Hello everyone, in this video lesson we are going to discuss about the process of translocation. In this video lesson we will discuss about the translocation process, where does it take place and what, let's say, contains the phloem sap. Then in another lesson or in another video lesson we are going to discuss about the process of loading and unloading the sucrose in sieve tube elements. Translocation, it is a term which is used to describe both transports in the plants, the transport that use the xylem vessel and the kind of the transport that use the Phloem vessel. Translocation, it means moving or transporting from one place to another. Translocation is a term which is mostly used to transport within phloem vessels, to transport the assimilates, which are the organic soluble compounds, which are transported through the sieve elements, which are elements or cells of the phloem vessel. So you can see in the picture, let's see the movement or the direction of the way in which, let's say, the assimilates move or are transported. So you can see that these are done with the help of xylem and phloem vessels, and this movement of the assimilates, it is done in two directions, from in here to, for example, from the leaves up to the roots, or in the opposite direction from the roots to the leaves. But first of all, let's see what structure does the phloem tissue have. Phloem, it is made up of sieve tubes, which are, and also the companion cells. Sieve tubes and the companion cells, which are cells that always associate the sieve tubes. Each sieve tubes, it is a living cells, which means that it contains, let's say, cell wall, cell membrane, cytoplasm, different kind of organelles such as endoplasmic reticulum and mitochondria. But let's say that there are no nucleus found in a sieve tubes, but these are found in a companion cell. The most important element of a sieve tube, it is the sieve plate. Sieve plate, it is the structure in which I did a circle, a blue circle. It is the place where the two cell walls of the sieve element join together, and this place or this structure which is formed, it is called the sieve plate. And sieve plate, it is made up of large pores which these are opened, and they allow the flow of liquids from one cell to the other in different kind of directions. So sieve plate, it is the place, let's say, where two sieve elements meet to each other, and these are opened pores which present a little barrier to free the flow to the free flow of liquids through them. The second type of the phloem vessel were the companion cells. Companion cells, same as the sieve tube elements, are living cells, but they do have a nucleus, as you can see in the picture in here. This is the nucleus. Companion cells are very metabolically active, which means that different kind of reaction takes place in there. They have numerous of plasmodesmata, which are, let's say, gaps between cells that allow the contact to the sieve elements. So here are the plasmodesmata, and these plasmodesmata, let's say, allow the movement of materials from the companion cell inside of the sieve element. Let's see What does the phloem sieve tube contain? The liquid in which the phloem, let's say, circulates, or the liquid which is found inside of the phloem sieve, it is called the phloem sap. Phloem sap, it is made up of different kind of substances, which we can mention those such as sucrose, potassium, amino acids, phosphate, ATP, auxins, which are hormones, etc. All these form what we know as a phloem sap. In this picture or in this table, it is shown, let's say, the solute found in the plant of Ricinus communis. The contents of the sieve tubes are in a very high pressure. In the moment then we cut, if this is a sieve tube, in the moment when we cut a sieve tube, let's say that the pressure which is very high inside will move toward the content, toward the cut. When the contents come up against the sieve plate, so if here we have the sieve plate and the contents want to go outside, let's say that the sieve plate might be blocked, and this helps to prevent the escape of the contents out of the sieve tube. The sieve plate prevents the content to go out by sealing itself, let's say, or the sieve plate, with a carbohydrate substance which is known as the callose, which works as a clot to prevent the moving of the phloem sap or the content outside of the, let's say, sieve tube. But how is it able, let's say, in nature to collect the phloem sap or to collect, let's say, the contents inside of the phloem vessels? You can see in the picture that this is a phloem vessels which contains different kind, let's say, of substances or solutes. And in here it is shown the ability of insects such as aphids, which let's say are able to collect the phloem sap through their tubular mouth parts, which are called stylets. These mouth parts allow the aphid to go inside and to, let's say, to collect the phloem sap without the damaging the phloem vessels, which means that the stylet parts of the aphid mouth parts are very small in diameter, and this allows, let's say, the aphid to collect freely and in a simple way the phloem sap, because this small diameter doesn't allow or doesn't trigger enough pressure, let's say, to make the phloem vessels to use the clotting mechanism. So the fact that the diameter of the mouth parts of the aphid it is very small, this doesn't surge, let's say, or trigger a high pressure of the content inside of the phloem vessels. Thank you.